Good morning and warm welcome to my channel. Today I discuss a very important and essential instrument that we have used in laboratory. Yes, this is a microscope and this is a compound microscope and another is a simple microscope. Today I discuss both of them, and first of all I must discuss the compound microscope, its part and their functions. So let's start. Before I start this video, I mentioned that please subscribe my channel and like this video. So without wasting your time, let's start. This is the compound microscope, and I discussed that this is the base of this microscope. This blue part of this microscope is a base on which total instrument is laid down, and after that this is the mirror. This is the plano-concave mirror. On which the light is come and reflected into the condenser, and after that it condensed into the objective lens. So this is the mirror, this is the plano-concave mirror, and it actually provides the light into the condenser. And after that, just above the mirror, this is the condenser.
The function of this part is condense the light that come from the mirror and ultimately enter into the condenser that reach to the objective lens.
So next is this is the white part of this microscope is arm, by which we can carry this microscope one place to another place. This is the arm actually, and this part is the basically heavy part of this microscope. This arm and the base is the heavy part of this microscope. So next is the objective lens. This is the objective lens, and this is the revolving nose pad. This is the revolving nose pad on which objective lens is attached with this nose pad. And there is so many objective lens, that is 10x. Here is the 10x, and next is the 40x, and so on. So what is 10x? What is 40x? That means the objective lens actually increase the size of the object or microorganisms 40 times larger or 10 times larger than their original size. This is the objective lens, and it actually increase the microorganism 10 times to 40 times than their original size. After that, this is the body. This is the body of the microscope tunnel on which the light comes into the eyepiece. This is the eyepiece, and eyepiece is always 10x. 10x eyepiece actually carry the information, carry the light and enter into the eye, that we can transmit, that we can see the total picture under the microscope. So this is the objective lens, and here we set the 40. Suppose we set the 40x here, and objective eyepiece is 10x. So 10 into 40, that means 400 times larger than the original size. We can see the my objective tissue or my objective microorganisms, whatever you can see. If we set here 10x, then just revolve this revolving nose pad and set the 10x, and this here we can set 10x and eyepiece is 10s. That means 10 into 10, total 100 times larger than the original size of the target tissue, target cell or target microorganisms. So you can see that 100 times, 40 to 400 times and so on than the original size of the microscope. Here, two important screw of this microscope here. That's the large screw, that this is called coarse adjustment screw, and beneath the coarse adjustment screw there is a fine adjustment screw. Actually, we can use this coarse adjustment screw for up and down the body of the microscope. Here is the mechanism. Just revolve this and up the body of the microscope, and just opposite side that go down to the microscope body. That we can use for the up and down this tunnel, this body, and we can set the exact resolutions of the our target organism. And here is a fine adjustment screw. What is their function? Fine adjustment actually clear the picture of the target field. You can use this fine adjustment screw for clearance of the field and the picture, so you can easily observe the minute parts of the target cell or target tissue. Next is the square, blackish square part. This is the stage of the microscope on which you can use it for slide rest, and here we can set the slide, and after that just use this coarse adjustment screw and set the resolution of the of your target field. Next, this is the clip of the on the stage. It is used for just set the slide on the stage. Without this, just slide is passed away or move to the on the stage. So please use this clip for fix your slide on the stage. So this is the compound microscope, and you can use this for minute resolution, minute vision of your target tissue or target microorganisms. Next one is, this is the simple microscope. This is the simple microscope, and here is a single screw. You can see this. This is the coarse adjustment screw, only the coarse adjustment, there is no fine adjustment screw, and there is a base of the microscope, and on the base there is a mirror that you can provide the light into the objective, into the lens. And here is a stage here, and this is the clip of the stage, and this actually fixes the slide on the stage. And on above the stage there is a eyepiece. This is the 20x eyepiece. Sometimes this is the 10x or 20x or whatever you can use. That means the 20 times larger than the original size. Here you can see the large specimen, large microscopic specimen. You can see easily this by use this simple microscope. Like you can use this for 96-hour cheek embryo or 72-hour cheek embryo. That is the slightly larger than the microorganism or tissue or anything. So you can use this for simple microscope vision. So you can use easily here set the slide, and then use this coarse adjustment screw just ups and downs the body of the microscope, and ultimately set the resolution of the total microscope. And this is the eyepiece. Here I use this for 20x, that means 20 times larger than the original size. So this at all the microscope and their parts and their functions. So friends, if you like this video, please like and share this video with your near and dear ones, and subscribe my channel.